Hi everyone, myself Dr. Punita Sharma, Assistant Professor, Department of Chemistry, School of Basic and Applied Sciences, Maharaja Agrawal University, Baddi. Today I will talk about the chromatography. The name of the topic is Exploring Chromatography. The content we cover in today's topic is the introduction to chromatography, principle, chromatographic terms, classification of chromatography and applications. Chromatography basically is a Greek word which means the writing in the color form. It is made up of by the union of two words. First is the chroma, other is the graphene. Chroma means color and the graphene means to write. First this technique has been employed by the Russian botanist which is named as Mikhail Svet. This technique is first developed in 1900. Initially, it is used for the separation of the plant pigment from the leaf of a plant. The leaf is crushed and the juice has been extracted, which is further subjected for the chromatographic. In this, we will use calcium carbonate as a stationary phase, and liquid hydrocarbon has been used as the mobile phase. The separated molecules in this technique initially was chlorophyll, carotene and xanthophylls. They all are the color compounds. These compounds has different color, namely green, orange, yellow. So on the basis of these compounds, the techniques has been named as chromatography. The collective, we can say that the chromatography is a collective term used for the set of laboratory techniques which we use for the separation of the mixtures. The mixture is dissolved in a liquid which we know as mobile phase, which is carried it through a structure holding another material called stationary phase. The various constituents of the mixture travel at different speed, causing the analytes to separate. The separation is basically based on the differential partitioning between the mobile as well as the stationary phase. Now we will discuss briefly about the terms which we will use in the chromatography. First is the analyte. Analyte is a substance which we will use for the separation. We can say it is a sample for our techniques. Second one is the chromatogram. Chromatogram basically is the visual output of a chromatograph. Eluent is the mobile phase. Eluate is the solvent or the mobile phase which carries analyte. Lastly, we have detector. Detector is basically an instrument which is used for the qualitative and quantitative detection of the analytes after separation. Here we have three figures. First represent the eluent stationary phase and the elutates. Second is the chromatogram, and third is the result of a detector. Next we will talk about the principle. Principle basically consists in this principle we have two things which we have to take into consideration. First is the mobile phase and second one is the stationary phase. As we all know that the mobile phase refer to the mixture of substance to be separated, dissolved in a liquid or a gas. Our mobile phase can be a liquid or as a gas, while the stationary phase is a porous solid material through which the sample contain the mobile phase percolate. The interaction between the mobile phase and the stationary phase result in the separation of compound from the mixture. Here is a figure which shows the separation through TLC, which is a thin layer chromatography. It is also chromatographic techniques which is used for the separation. Now we will discuss about the general step which we will use or which we can use for the separation of different analytes using chromatography. First of all, we will have a stationary phase on which the adsorption or retention of the substance is done. Second is the separation of adsorbed substances by the mobile phase. Our adsorbed substances will dissolve into the mobile phase and start moving. Third is the recovery of the separated substance by a continuous flow of the mobile phase, the method being called elution. Next and the last step is the quantitative and qualitative analysis of the eluted substances. Now we will talk about the classification of the chromatography. Various techniques has been classified on the basis of their mechanism of separations, either on the basis of phases or in the basis of the shape of the chromatographic bed. First, on the basis of mechanism of separation, we will classify the chromatography as adsorption chromatography and the partition chromatography. Second, on the basis of phases, we classify the chromatographic techniques: gas solid chromatography, liquid solid chromatography, liquid gas chromatography. Third, on the basis of chromatographic bed, we will subdivide the chromatographic techniques into the column chromatography, paper chromatography, and thin layer chromatography. Next, first of all, we will discuss the classification of the chromatography on the basis of mechanism of separation. As I earlier told you that the separation mechanism also play an important role on the process of separation. First is the adsorption chromatography, second is the partition chromatography. In the adsorption chromatography, the separation is basically done on the basis of relative differences in the absorption of the component to the stationary phase. It is the oldest type of chromatography. In this, the equilibrium between the mobile and stationary phase accounts for the separation of different solutes. In this adsorption chromatography, we utilize a mobile phase or a gaseous phase that is adsorbed into the surface of the stationary solid phase. Here we can see the figure in which a solid stationary phase is there. On the surface of this stationary phase, there are analytes or the sample compounds has been adsorbed, which has been separated with the help of mobile phase. Next is the partition chromatography. In this, basically the separation is done on the basis of difference between the solubility of the component in the mobile as well as in stationary phase. A thin layer is formed on the surface of solid support by a liquid stationary phase. Here our stationary phase can be a liquid or a gas. Next is the classification of chromatography on the basis of the phases. First is the gas solid chromatography, second one is liquid solid chromatography, and third one is liquid gas chromatography. This is the instrumentation picture of this gas solid chromatography. In this, our mobile phase is a gas which is known as a carrier gas. The carrier gas is basically an inert gas that can be halogen, nitrogen, hydrogen, etc. The separation is basically depends on the relative partial pressure of the sample component above the stationary phase. In this, the solute equilibrate between the mobile phase, which is a gas, and a stationary phase, which is basically a solid support. This technique is used for the separation of atmospheric gases and volatile compounds like essential oils. Now we will discuss about the liquid solid chromatography. Mobile phase is a liquid. Separation is basically done on the basis of properties of classes of compound. For example, alkene compounds can be separated by using alcohol as a mobile phase. The preferred mobile phase is a non-polar or slightly polar solvent. The molecules has been carried out either in a column with the porous adsorbent, which is polar in nature. The popular adsorbent used in the liquid solid chromatography are silica and alumina. For example of liquid solid chromatography is the HPLC. Here we can see the representation of a HPLC system. Next is the gas liquid chromatography. In this, the mobile phase is a inert gas that can be a hydrogen, nitrogen, and the inert and the stationary phase comprises of a small amount of liquid held on a finely divided inert solid support. This technique is very sensitive, which is used for the detection of small quantity of substances. The stationary phase used in the GLC are alumina or silica. Based on the shape of chromatographic bed, we will classify the chromatography in the column chromatography, paper chromatography, and thin layer chromatography. Paper and planar chromatography. In this, the stationary phase is present on a plane, either in the form of a paper or integrated by a substance to as a stationary bed, as in the paper chromatography. But it can be present in the form of a layer of solid particle spread on a support such as glass plate or a alumina plate. There are different compounds in the sample mixture which travel different distance according to the affinity, how strongly they interact with the stationary phase as compared to the mobile phase. The specific retention factor RF value can also be used as an aid to identification of the unknown compound. Now the column chromatography. Basically in this we will use a column. Column is a tube shaped object. Here you can see that in the picture. It is the stationary bed is present in the column. The particles of the solid stationary phase or the support coated with the liquid stationary phase may fill into the hole inside the volume of a tube, which we call column. The difference in the rate of movement through the medium are calculated to the different retention time of the sample. Here in this figure we can see that in the first figure we will see there is a packing of column in which the silica is added as a stationary phase, and above the silica there is a addition of some slurry which contain our analytes or the mixture of compounds which has to be separated. As we add the eluent or mobile phase to the column, the separation process begins. The separation is done on the basis of affinity. The compound which has strong affinity is adsorbed strongly, and they eluted later on, while the compound which has less affinity, they will eluted out earlier. Now we will talk about the thin layer chromatography. In this, the sample's relative affinity towards the mobile and the stationary phase is the basis of separation. The process begins here by moving the mobile phase over the stationary phase surface. During this moment, the higher affinity compound gains less speed as compared to the lower affinity compound. Different spots can be found on the stationary surface at distinct level, reflecting various elements of the mixtures. Now we will talk about the application of the chromatography. Chromatography techniques have wide application in different fields. In the pharmaceutical industry, we use chromatographic techniques for the analysis and identification of the presence of any trace compound of chemical and elements in a given sample. We can use this technique for the separation of certain chemical compounds based on their molecular masses. Chromatographic techniques play a crucial role in the development of a new drug, and it will keep a check on the quality of the drug. The presence of impurity and other unknown compounds can be detected in the drug sample. Now in the food industry, it is used for the determination of shelf life of the food substance by helping in the analysis of the point at which our food gets spoiled. It also used in the to check the presence of chemical additives in the food. Next, it is a technique which we can use for the evaluation of nutritional value of our food. In the chemical industry, it is used for the testing of water sample, testing of air samples. The presence of toxic component can also be detected by using these chromatographic techniques. It can also be noted that the chromatography has many applications in the life sciences. In the field of life sciences, we used it in the studies of proteomics, metabolomics, and protein analysis. Nucleic acid research is also known to make extensive use of chromatographic techniques. HPLC is widely used in the protein separation. It is also used in the enzyme purification, plasma fractionation, and insulin purification. These are few references which you can use for the further studies.